Hello everyone, myself Dr. Shweta Kamboj. I have done my PhD in microbiology from Punjab University, Chandigarh, and currently I am working as an assistant professor at Department of Biotechnology, CGC Landra. So today we are going to discuss a very important topic of molecular biology, that is translation. So before going into the detail of the translation process, let us discuss what is central dogma. So a central dogma is basically what? We have a double-stranded helical structure of DNA, and this double helical structure of DNA has the ability to replicate its own self by the process which is known as replication. After which this DNA get converted into a single-stranded structure which is known as RNA by the process transcription. After this RNA get converted into the protein by the process known as translation. So this whole process is known as central dogma, and out of this today we are going to discuss the translation. So before going into the detail of the translation process, there are certain requirements for the translation process. There are certain factors, certain molecules, certain structures which are required for translation process. So one of them is codon. There are two types of codons. One is start codon and one is stop codon. So as the name indicates, start codons are those codons which start the process of translation. And what is translation basically? Translation is the synthesis of protein from the RNA. So the start codon which is used is AUG. Now the stop codon, as the name indicates, are the codon which stops the translation process, that is protein synthesis, and there are three types of stop codon, that is UGA, UAA, UAG. So in addition to this, we have certain structures which are very important for the translation process, such as mRNA, tRNA and ribosomes. Out of this, the ribosome is one of the most important structure in the process of translation, as it is a site where the protein synthesis occur. So ribosome is basically made up of two subunits. One is the larger subunit and another one is the smaller subunit. The larger subunit is known as 60S and the smaller subunit is known as 40S, and these two units collectively are known as 80S, and which collectively play an important role in the process of protein synthesis. So in addition to this, these sites of ribosome has three sites, which is A, P, E site. A stand for amino acid, P stand for peptidal and E stand for exit. And all these three sites plays a very important role in the process of translation. So the process of translation is basically divided into three steps. First is initiation, then there is elongation, and the last step is the termination. So before the process of translation starts, that is before their initiation process starts, first there is a requirement for the activation of amino acid. So as we all know that proteins are basically made of amino acid, or we can say that amino acids are the building block of protein. So for protein synthesis to start, there is very much requirement that the amino acid which are going to take part in protein synthesis are activated. So the enzyme which is responsible for the activation of amino acid is amino acyl tRNA synthase, and in the presence of ATP, this amino acid gets activated, and after activation it gets associated with a charged tRNA molecule, and which take part in the process of protein synthesis. So there are certain factors which are involved in translation process. Each factor is specific for each process. So as for initiation, we have IF1, IF2 and IF3. Here IF stand for initiation factor. So initiation factor binds to the A site, initiation factor 2 brings the first tRNA to the P site, and the initiation factor 3 keeps the two ribosomal subunit dissociated. And in the elongation process, we have another two factor, which is EFTU and EFG. Here EF stands for elongation factor. The EFTU role is to bring the tRNA to the A site, and EFG cause the translocation. In the next step, we are going to study them in detail. The last step is the termination, for which we have factor RF1 and RF2. Here RF stands for release factor. So the RF1 works when we have stop codon UAA, UAG, and RF2 works when we have the stop codon UAA and UGA. So now let's discuss each of this process in detail. So for the initiation process, the amino acid which got activated and get associated with the charged tRNA comes, and after this there is a mRNA molecule which have a start codon as we discussed earlier, that is AUG. This mRNA and two ribosomal subunit join together and a tRNA with activated amino acid is bring to the site P by the factor IF2. Now the elongation step. So as we have seen in the initiation step that the first amino acid along with the factor IF2 come to the site P. Now as we see that A site is empty, now the next amino acid, activated amino acid with the tRNA will come to the site A. After when both P and A site are occupied with the tRNA molecule and the amino acid, there will occur the bond formation, and in case of proteins, the bond which is basically formed is the peptide bond. So the factor which is after the formation of bond, the factor which is responsible for the translocation, that is EFG, comes and it causes the translocation. So what basically is translocation? As you all can see that here the tRNA is present at A site and then the P site. So what in translocation occur? Once the bond is formed between the two amino acid, there occur the shifting of these tRNA from one side to the other. So as you see here, the tRNA was present on P site. This tRNA from P site will go to the E site, and from A site the tRNA will go to the P site. So here again the A site is empty and this A site is ready to receive another tRNA molecule. So here we can see that the site, A site, which is empty, has received a new amino acid molecule. Again, there will be the formation of the peptide bond formation. As you can see that there will be a formation of bond between these three amino acids, and after the formation of these bonds, there will again the translocation, and in translocation again the tRNA which is present on the P site will be shifted to the E site, and the tRNA which is present at the A site will be shifted to the P site, and the molecules which are now the tRNA molecule which is present at this E site will exit through it, and in this way this process keep on continuing. So this figure also shows the same, that once the tRNA molecule come and there is a formation of the peptide bond between the amino acid, there will be the translocation, and again the A site, every time the A site will be empty and it will be ready to receive the next amino acid. So the next step is the termination of translation. So once there is a formation of a long polypeptide chain, the release factor, that were RF1 and RF2, they encounter the stop codon. So basically there are three types of stop codon: UAG, UAA and UGA. So depending upon what type of stop codon the release factor encounters, if it encounters UAG and UAA, release factor 1 will work, or if it encounters UAA and UGA, release factor 2 will work. So as the release factor encounters with this stop codon, the process of translation stops here only. And after, as the process stops, there will be the release of the free polypeptide chain, which will undergo further process of maturation for a synthesis of a mature protein, and rest of the structures which were initially joined during the initiation process will get dissociated, such as the two ribosomal subunit will be dissociated, mRNA structure will be dissociated, and tRNA molecule will also be dissociated, and in this way we get a free polypeptide which will undergo maturation. So as we see that from as soon as the release factor encounters the stop codon, there is the release of the free polypeptide chain. So this is the process of translation, which was divided into 3 steps: initiation, elongation and termination. I hope you liked this video and you are able to understand. Thank you everyone.